Exam video number 5. So as you can see here, we have the template strand and the coding strand on the bottom, but we want to read it into the 5 to 3' direction, so we are going to flip it. So as you can see here, this is the beginning, and then we are going to try and transcribe it into mRNA by changing all the T's to U's. And so when we do that, we see our start codon here and our stop codon here. And this codes for methionine, valine, arginine, thymine, and lysine, and then the stop codon. And how do we know what are the stop codons and what are the start codons? So I remember this by school starts in August, so AUG, which codes for methionine. And the stop codons are you are annoying, you go away, and you are gone. The type of point mutation. So a silent mutation is when one amino acid is coded for, and when you change the base pair, the same amino acid is still coded for, and this is largely due to the wobble effect. A read-through is when a stop codon is changed to form an amino acid. Missense is when an amino acid changes to another amino acid. And nonsense is when an amino acid becomes a stop codon, and this results in a shortening of the DNA. Transition is going from purine to purine or pyrimidine to pyrimidine, and transversion is pyrimidine to purine or purine to pyrimidine. How do we know which ones are which? Well, I'll tell you. It is purely adorable when babies say GA, and that stands for guanine and adenine, and then pyramids are sharp so they can cut you, and the CUT stands for the cytosol, uracil, and thymine. And as you can see here, it went from a C to a G, which would be a transversion, and then GUC coded for valine. GUG also codes for valine, and so this is a silent mutation. This here, it went from an A to a C, which is transversion. UAA coded for stop, UAC codes for tyrosine, and because of that, it is a read-through mutation. This next one, we have G to C, which is also a transversion. AGG coded for arginine, and AGC codes for serine. So it's 2 amino acids, but they are different, and so that is a missense mutation. And finally, we have A to T, which is also a transversion. AAG coded for lysine, and UAG codes for stop. So this is going to stop right here and shorten this DNA sequence. Nonsense mutation.